family. You chat, you eat lunch, and overall you have a great time. Unknown to you, however, is that the deli meat you had on your sandwich was contaminated with bacteria. Depending on the species, you might experience some nausea or gastrointestinal discomfort. And while that's definitely annoying, you'll be able to resume your everyday life soon afterwards. But what if those bacteria could spread from your gut to your brainstem, causing disease in your brain, all from a piece of contaminated food? Now, instead of being nauseated, you might have a headache, swelling of your brainstem, and face potential loss of motor function. The bacterial pathogen I work with, Listeria monocytogenes, is the third leading cause of death from foodborne illness in the United States. This pathogen poses a huge risk for the food industry because unlike many other bacteria, it can continue to replicate even at low temperatures, like those found in your refrigerator. Listeriosis, the disease associated with this pathogen, is relatively rare, but 20 to 30% of people that get it die as a result. It primarily affects those with weakened immune systems and pregnant individuals. Even scarier though, there are certain strains of this bacteria that can spread to your brainstem, even if you are young and otherwise healthy. We refer to these strains as neurotropic, which simply means that they're able to invade tissues of the nervous system. Our lab has previously found that these strains make it to the brain without becoming abundant in the bloodstream. So we think that some event in the gut allows these bacteria access to the brain. Our hypothesized route of dissemination, as shown on the left side of the slide, is that as food contaminated with Listeria passes through the lumen of the small intestine, the blue area of the box, some of these bacteria, shown as green structures, are then able to cross the protective layer of intestinal epithelium by first moving into these cells and then exiting out the other side into the lamina propria, the beige area of the box. We found that these strains do not invade neurons very well, but they can invade enteric glial cells about 2 to 3 times more efficiently than other Listeria strains. Enteric glial cells, shown in yellow, surround, support, and are in direct contact with fibers of the vagus nerve that innervate the gut. So, we think that these strains first invade into these enteric glial cells, and then utilizing cell-to-cell spread, a mechanism Listeria frequently uses to move throughout the body, they spread from these glial cells directly into the adjacent neurons. Once there, they can travel intracellularly all the way up the vagus nerve until they reach the brainstem. Invasion into specific cell types is mediated by bacterial surface proteins. As you can see diagrammed in the upper right, these proteins interact with components on the surface of the target cell, promoting entry of the bacteria into the cell. Our lab has found that there are 14 surface proteins that are in these neurotropic Listeria strains that are not found in non-neurotropic strains. Of these 14, 5 have structural similarities to known invasion factors of Listeria. These factors allow Listeria to invade into intestinal epithelium tissue, like you saw previously in the box, or into cells in your liver. So we think that one of these 5 similar proteins might confer the ability to invade into enteric glial cells so well. But my project has focused on taking one of these 5 proteins and creating a mutant where the gene that encodes for it has been deleted. The protein I've been investigating is referred to as 1096. Once I created my deletion mutant, I've now been testing it against its parent neurotropic strain to see how the two compare. To do so, I have conducted experiments where I have a cultured enteric glial cell line, and I add these Listeria strains to the cells. Then, I quantify how many of those bacteria were able to make it inside of the cells during an hour-long incubation. As you can see on the bar graph in the bottom right, the loss of this protein causes a reduced capability of these strains to make it inside these enteric glial cells. The y-axis, percent invasion, is a measure of how many bacteria we recover from the inside of these cells versus how many we added to the dish in the first place. The bar on the far right, which represents the strain that has had gene 1096 deleted, no longer acts like its parent neurotropic strain. This loss of the protein 1096 makes it basically incapable of invading into these enteric glial cells at the same level it once did. Bacteria have a whole host of mechanisms that they can use to move throughout our bodies and to cause diseases. And by better understanding these pathways, we can target them for therapeutics and for treatments of these deadly infections. Thank you for your time.
Thank you, Tanner. Very clear, and even for me. So A question occurs to me, though, in terms of therapy, therapeutic use, that is. How would a consumer, one consuming food, how would you know you need this? How quickly do you hypothesize that the Listeria is going through the gut and up into these cells? Would a patient have enough time to know, ooh, and take a Tums equivalent of your 1096 and away you go?
Yeah,
I guess it would be something that would cause that to happen, but.
Yeah, that's a fantastic question. We're not exactly at that stage yet, and unfortunately, the issue with Listeria is that frequently you don't know whether you have consumed that bacteria until later on. And even more so, these strains that possess neurotropic capabilities are from a genetic lineage that have not been studied very well in the Listeria field, so we don't know that much about them at all. In terms of our mouse model, they typically make it to the brain about 4 days post-infection. So potentially some sort of therapeutic could block the majority of bacteria getting to that point. But it would take a lot more research to figure out exactly what symptoms might occur in the early stages of that infection and being able to predict whether that infection would actually progress to the brain or not.
Okay, first of all, now I'm scared. I'm going to go home and clean out my refrigerator. So what is the current standard of care, and has there been any comparison to that in terms of treating Listeria?
So the current way that we treat listeriosis in patients is just with normal antibiotics. The primary issue is that, as you can see from the hypothesized route of dissemination, these bacteria spread primarily intracellularly, and antibiotics aren't able to cross that cell membrane to make it inside of our cells. So Listeria infections are frequently resistant to antibiotics, which is why we need to find some new method to help treat these deadly infections.
How did you knock out protein 1096?
It was through homologous recombination. So essentially, I took a plasmid, which is just a circular piece of DNA, and I included the regions of the genome that are upstream and downstream of the gene, and then transformed that into Listeria. And so transformation is just adding the plasmid into the cell. Then by promoting recombination events, I could force the plasmid to integrate, and then it would loop out, and half of the ones that looped out would either have the gene left and half would have it gone. But I added, we essentially confirmed by genetic sequencing to make sure that the gene was deleted. And the results that we got, this gene was the only thing that was altered in the chromosome of this new mutant strain. Mm-hmm. Thank you so much.